3:30. So it's my pleasure to introduce our opening speaker, John Coburn. He's a professor at the University of Birmingham and also a director of the Center of Environmental Research and Justice. And today he will talk about multiomics through the lens of evolution. So maybe if John is ready, you can introduce yourself and talk a little bit about what you are going to bring us today.
Thank you very much, Maria. And thank you all for having organized this fantastic seminar series over the next 2 days. I can't believe how many people have lined themselves up to enjoy this phenomenal lineup of speakers. My name is John Coburn, and as Maria had said, I am a professor at the University of Birmingham, but I also direct a large project, an EU-funded project called Precision Talks. The outcome of this project is very much aligned with the goals of this symposium and workshop. And I hope that through this discussion, we can basically have more dialogue around where this project fits in, generally speaking, in our attempts to advance the science in terms of mechanistically understanding the threats that wildlife and humans face with respect to chemicals in the environment, and what the science can do in terms of mitigating those effects in all kinds of different areas of governance. So I will share my screen if you don't mind. And I hope that you can see my first slide.
Now we can see it.
Great, thank you very much. So as I said, the project is called Precision Talks, and it involves 15 organizations across 8 countries. And the goal of the project is to improve chemical safety assessment, to better protect human health and the environment by using non-traditional test species, meaning that they're non-sentient and abiding to the 3 R's principles, multiple fields of knowledge, powerful computational approaches to understand which chemicals are toxic and why. And the panel of species that we're going to be dealing with during the seminar are invertebrate species, Drosophila melanogaster, the premier biomedical model species for genetics, Daphnia magna, a premier ecotoxicological model species. We're also doing work with C. elegans, the embryos of Danio zebrafish, the embryos of Xenopus the frog, as well as a human cell line. Now, the core research is comparative toxicology. What we're dealing here with is the notion that the responses that organisms have to the harmful effects of chemicals would be shared among species by evolutionary descent. And why should they not? Most of the phenotypes that we have as humans are in fact not invented by humans. They're derived by our common ancestry with other mammals and other animals in general. So the notion that we have in terms of precision talks is to try to understand the evolutionary origins of toxicity pathways. Now, this is important because I think that, generally speaking, regulatory toxicology is moving away from concentrating our efforts on observing adversity through the toxicological effects on survivorship or reproductive success, but instead try to understand and regulate the toxic effects of chemicals based on their modes of action. So the way that we're doing this is that we are exposing these model species to 200 chemicals, and we are observing their phenotypic effects in all kinds of different areas, whether they're behavioral or they're morphological or death, reproduction. But we're also extracting from the samples the metabolites and the RNA, therefore representing the biomolecular responses of these organisms, in order to use machine learning approaches that reconstructs what we call these molecular key events. And the idea here is that if we observe molecular key events that are discovered through this process among a subset of the animals, in this case, we have the observations in the vertebrates, but not the invertebrates. Presumably, this would mean that the pathway to toxicity had evolved somewhere along the branch leading to the vertebrates. And this facilitates our attempt to do cross-species extrapolation, because presumably, if that biomolecular response, the key event, evolved at the base of the vertebrate phylogeny, you would at least begin to hypothesize that it would affect all vertebrate species. And conversely, not have a great effect on the invertebrate species simply because those invertebrates do not actually have that toxicological pathway. And so we're doing this through large-scale data acquisition and analysis. We're doing comparative transcriptomics using RNA sequencing machines and comparative metabolomics using mass spectrometry. The diversity of chemicals is quite large. As I said, we're using 200 chemicals, but we've chosen them very carefully to hit a variety of different toxicological outcomes or pathways. In this case, among the 200, we see 22% of them are known to be hepatotoxicants. Now, this is very interesting because not all of our model species actually have a liver. So it's a very good hypothesis driving set of chemicals that are being used for this study. There's some genotoxicity, some hematotoxicity, some immunotoxicity as well. But also, many of the compounds that we're using, we do know something about their mechanisms of action. And in some cases, they're actually nuclear receptor mediated. And so one thing that we've discovered by looking at our chemical library is that in many cases, many chemicals actually have many different modes of action. And so we're hoping that we're going to be able to extract this kind of information from our assays. The first thing that we need to do is we need to set some sort of criteria as to what concentrations do we expose these animals to. And so they're all basically being tested in a dose-responsive manner. And here's one example of our model species being tested against increasing concentrations of acrylamide. And you can see right away that the dose response curves are quite distinct from one another. Daphnia seems to actually be the most sensitive species for this substance at 2,000 micromolar, hitting the base level dose toxicity of 50%. Drosophila melanogaster is much higher. And also the shape of the curves are quite distinct. Daphnia has this very nice sigmoidal curve that's shared with Danio, but if you look at the Xenopus embryo, it basically is kind of like jumping off a cliff. It's not toxic until it is, and when it is toxic, it kills 100% of the test organisms. So once we've actually determined the base mark dose of these substances against all of these species, then what we can do is we can begin to understand their relative sensitivities. Now here what I'm doing is I'm plotting for each of the model species the rank order of the substances that have been tested as a matter of concentrations of their BMD50s. And once again, as we had seen with acrylamide, Daphnia happens to be the most sensitive of all species, where for each of the substances, they respond toxicologically at a much lower dose than, say, Danio or Xenopus. You can see that the nematode worm is much resistant to many substances, and Drosophila even more. There's a lot of biology here, of course. Drosophila, for instance, these are adults and they're being fed the substance. And so the way by which these substances are being absorbed into the organisms are very different. And you would see this as a biological effect on this graph. But the important thing to note is that based on a non-parametric Kendall-Tau test, the relative toxicity of these substances are consistent across all of these species, more or less. The more closely related the species are, the more consistent is the rank order of the toxicity of the substances. But that's important because that means that the way by which these toxicants are affecting the organisms may have different sensitivities, but relatively speaking, they actually share the same toxicities to one another. There's another way of looking at this. Here's a figure of the BMD values, the benchmark dose at 50% for each of the test organisms across each of the substances, looking for their correlation. No surprise, if you compare Drosophila females to Drosophila males, the correlations are quite high. They're sitting around 98% correlations for each of the tested compounds. Daphnia versus Drosophila, the two arthropods, also have a very high relationship in their responses. But what's quite stunning here is that if you look at the invertebrates, on average, they correlate with one another in terms of their responses toxicologically to these substances, around 76% correlation. But if you start comparing now between the vertebrates and the invertebrates, you get a number that's not so different at 68% correlation. So in general, the organisms are in fact quite similar to one another, despite the fact that their evolutionary descendants goes back to last sharing a common ancestor more than half a billion years ago, so during the Cambrian explosion. Now, what we want to do in terms of comparative toxicologists is that we really want to understand the evolutionary origins of the toxicity. And here's one example of now plotting this data for a substance called cyclosporin A, where at the end of these branches, you're seeing what their benchmark dose at 50 was measured at. And in this particular instance, there was no toxicity that was observed among the invertebrates, but there was a toxicity observed among the vertebrates. And by doing phylogenetic reconstruction of toxicity as a trait, you can use likelihood analyses and other methods to infer what the toxicity was of the descendants of some of these species. These would be the internal nodes. And so in this case, it's an example of toxicity that has obviously an effect on only the vertebrates, so therefore has presumably a toxicological pathway that has evolved at the branch to the vertebrates, but not to the invertebrates. And again, one has to appreciate the evolutionary distances among these species. To find such similarities among them is quite stunning. Now, this is a workshop around omics, and multi-omics is the key. And so what I'd like to do is to begin to share with you the data that we're getting for a pilot study of 6 substances, starting with first comparative metabolomics, then going to comparative transcriptomics, and then a multi-omics investigation of the data. And again, the purpose here is to discover in a data-driven way what the toxicity pathways might be, and by comparing them across a species, be able to infer at what time during animal evolution these pathways emerged as being relevant. Now, comparative metabolomics is rather new. I think that everybody on this call would be quite familiar with comparative genomics that started when we started sequencing the genomes of a whole bunch of species and being able to compare the gene sets and the genome structures among them. But comparative metabolomics is quite new, in fact, and part of it has to do with the fact that the data needs to be pretty homogeneous. And in this case, if you imagine what we're dealing with here, we're doing a very standardized, very controlled experiment across all these model species. So we're trying to eliminate as much of the technical variability as possible and focus our attention on the similarities or differences that we see from the data because of biology. And this processing workflow is designed for this project, where we end up taking the metabolomics data. Typically, these are mass to charge ratio data. We detect the peaks and the alignments that are consistent across the biological replicates. We create peak intensity matrices. We have a multi-step signaling, filtering, and signal correction pathway to then normalize the data, do the missing value imputation, transform and scale in order to begin to do both univariate and multivariate statistics on the data. One of the main difficulties with metabolomics is the true identification of the metabolites, because really what you're looking at here is a fingerprint of roughly 8,000 to 12,000 peaks that represents all the endogenous metabolites in the organism. And some of those peaks can actually be identified because there are standards that are included within the assay. And so you can identify those peaks that are tied to those standards in this way. And so some of the compounds are identifiable at very high accuracy. Others can actually be putatively annotated based on physical chemical properties, known characteristics of the substances, and imputing based on what would be the presumed mass to charge ratios of those peaks, be able to identify metabolites in that way as well. But the majority of them would be putatively characterized compounds by statistics, or most of it would be unknown. So we have to concentrate our efforts right now on only those that are identifiable across the species. And so what we have done so far is that by looking at the data across the two different concentrations of 6 substances at 5 different time points, we were able to positively identify between 359 and 580 known metabolites across these model species. And so that's the graph to the left here. And these would be a very, very high confidence. And the graph to the right is showing the degree to which these identifiable metabolites are shared among the species. And among the 1,107 total annotated metabolites, they contain 1,430 compound names. The largest class is a class of 168 substances or metabolites that were identified in each of the model species, including the human cell lines. But many of them are actually uniquely found to each of these species. So for instance, Xenopus, there's 106 of them that are only found in the experiments that included Xenopus. But gratefully, there are some that are shared uniquely, let's say between the males and females Drosophila. That's comforting. Some of that are found, 21 are found uniquely among the arthropods, so the two Drosophila sexes and Daphnia. But yet again, look at the differences that you see even between the sexes in Drosophila, where 79 of them are identified only in females, whereas 64 are identified only in males. So now that we can actually compare metabolites as a function of the treatments that these organisms received, is there any signal that we're getting from the metabolome of a toxicological pathway or toxicological response that's tied to the pathway? And I'll give you 4 examples here, simply comparing Daphnia to Drosophila. Again, they may be 2 arthropods, but they have last shared a common ancestor, hundreds of millions of years ago. And so there's quite a bit of biology here represented that's unique to these organisms. And sure enough, when you expose them to cadmium chloride or pyridoxalic acid, erythroprophis, or sodium arsenite, you get to see that the majority of the metabolites that were identified in each of those substance exposures are unique to either one of the 2 species. But there is some overlap. There's a 10% overlap of metabolites for cadmium chloride, 7% for pyridoxalic acid, which is a PPAR agonist. Erythroprophis, there's a small overlap as well, and sodium arsenite. And what I've then did was basically ask the question, well, is there something special about the metabolites that are overlapping? And the answer is yes. A few years back in 2022, myself and with colleagues, including Professor Mark Viant, had completed some work that we've done as a group with the European Chemicals Agency at putting together and creating something that's called the MTOX 700 metabolite panel, very similar to the US EPA's toxicological panel that is being used for transcriptomics. And I believe that Dan Villeneuve or others are going to be speaking about that either today or tomorrow. And if we look at basically those metabolites, and you ask the question, well, are they binned in any way as being unique to either the 2 species or the shared function of the 2? They are overwhelmingly overrepresented in the set of metabolites that are co-expressed between the 2 or co-detected between these 2 species for each of the 4 substances that are shown here. And so for cadmium chloride, for instance, we get to see that 37 or 78% of the 37 metabolites are in fact members of this MTOX 700 metabolite panel that are predictive of a toxicological effect, 87% for pyridoxalic acid. So that's quite comforting because it's basically indicating that this comparative method is quite capable of dissecting out from the overwhelming biology that is represented within these data sets that are part of a living organism, whether it's Drosophila or Daphnia. It's the overlap that when you capture that signal is truly the most representative of the actual exposure scenario, the exposure responses of these organisms. Some interesting things that can be actually seen and observed from the biology itself. If you compare, for instance, the effects of time point on the metabolome, I'm showing you here these 5 time points being exposed and the metabolic changes that are happening, including all the substances and both the concentrations that were tested. And you can see that for the 4 organisms that are developing during the course of a 48-hour exposure, there are some differences in terms of metabolic changes that are quite abrupt. Here for C. elegans, you can see that there's an abrupt change in the metabolomes going between time point 3 and time point 4. For Daphne, it's between time point 1 and time point 2. For Denio, it's between 2 and 3, and there's another one here for Xenopus between 1 and 2. Now, that could make a lot of sense, of course, because during development, there's different biologies that are occurring, and these toxicants are in fact interfering with the biology at each of those time points. And so something must be happening developmentally that is affected by the substances that you do not see if you're doing, for instance, the Drosophila adults. So they no longer are developing during the experiments. The males here do have something that's interesting going on between time point 4 and 5, but there's nothing significant really even with the human cell lines. Now, is this generally speaking a response or reflective only of development? And the answer is no, because if you now look at the time point chemical interaction of the metabolome, now we're basically dissecting out how the metabolome is changing for, let's say here, nematode worms and Daphnia, and you compare them across the different substances, you can see that it's Erythroprophis that's driving this change between time point 3 and time point 4. Erythroprophis is also driving a metabolome change in Daphnia between 1 and 2. So obviously what Erythroprophis is doing is that it's interfering with something very specific about the biology of these two organisms at a very distinct point in their development. Pyranixylic acid is doing something in Daphnia that we're not seeing in other species. And again, this is being the changes that we're seeing during the development is in fact very much specific to specific substances as a function of their modes of action. And finally, what's really interesting is to look at things like, for instance, the differences between the sexes. So here we're just comparing the female and male Drosophila, and a linear modeling indicates that there's a huge difference in the metabolomes that are purely driven by the sex of these organisms. But as you can see, as well as you look at the metabolomes changing over the time points for each of the substances, the changes that are occurring in the females are far more wild across the different time points for each of the substances than it is compared to males, which is a lot more stable. So we are seeing a lot of biology from these metabolomic data sets. And I hope that what I've been able to show you here is that, first of all, it's interesting to be able to do these comparisons because of the controlled nature of the experiments, but that you can actually tease away some signals here that are specific to how these organisms are responding, toxicologically speaking, to the exposure of these substances. I'd like to now move on to comparative transcriptomics. And again, just to reinforce the nature of these data sets, we're doing this on 200 substances, ultimately 200 substances at one concentration and 2 time points. But this initial data set that we're looking at today, we have all these genes that are being interrogated, 2 different dosages, 5 different substances, 5 different time points. This simply leads to interpretation fatigue, and this is just a very, very small subset of the total data that's being collected. So how do we deal with that? How do we able to extract meaningful information without having to analyze every single change? Well, we've applied an algorithm here in collaboration with our colleagues at the Oak Ridge National Laboratory in the United States called MENTOR, stands for Mechanistic Exploration of Networks for Team-Based Omics Research. And what this does is it functionally partitions the feature space of these transcriptomes, and they're embedded within these pre-trained projection models that are derived from a huge volume of prior human knowledge. So what we're doing here is that we're using our test species, whether it's a sentinel like Daphne or Drosophila, and we interpret the gene response changes as orthologs to the genes that are shared with humans. And so we're kind of like treating all the transcriptomes of these species as if we're actually observing human genes as opposed to species-specific genes. We look at the omics results, so the differential expression patterns that are happening across the different treatments, and we compare those against what is called the multiplex. We're doing some random walk embedding across all the known interactions that we have in humans derived from data sets that include the HumanNet version 3, the ENCODE transcription factor target networking, or the GTEx predictive expression networks. And so what we're doing is that we're basically asking the question, given all the knowledge that we have of the interactome in humans, can we detect some associations within the transcriptomes in each of our model species that would be occurring less likely by chance, but driven by the fact that that in humans at least, we know that these genes interact in particular networks and functions. And ultimately, this leads to what is called a hierarchical clustering of embedded gene classes. And what we do, therefore, is each of these groups of genes that are found to be co-regulated against a human a database of interacting networks, we then are left with having to interpret those and ask the question, what do those function? And we can divide up the task by simply just assigning different groups to different sets of gene clades. The results are quite stunning. Here, I'll just give you one example. Here's the transcriptome of comparing Drosophila to Daphnia. And what, as you've seen from the prior slide, we're creating basically human knowledge networks of genes from the transcriptomes of these two species. And in the case of sodium arsenite, we were able to identify 100 clusters or 100 groups of human interactome elements that are shared between Daphnia and Drosophila. So presumably speaking, what we're seeing here is that whatever we know about the human interactome, we're observing it in Drosophila and Daphnia, and we're observing it in a treatment-specific manner. These are observed when they're both exposed to sodium arsenite. And the thing that really strikes me right off the bat is that although we're able to pull out these human networks from these two species, in green are those genes that are orthologous to humans that are differentially expressed in Daphnia, and in blue are the ones that are differentially expressed in Drosophila. There's barely any overlap in those genes. In fact, 8% only of the co-regulated human known gene networks between these two species are being identified by genes that are changing or differentially expressed in both of the organisms. And so again, it's telling us a little bit more now about the preservation, the evolutionary preservation of response genes or response networks, in that the network seems to be very much preserved, humans, Drosophila, Daphnia, but the elements within that networks are not necessarily the same ones that are responding in a differentially expressed way. Now, let's move on to how we deal with the multi-omics. And in this case, we're applying an algorithm that's been developed by a colleague at the University of Birmingham. His name is Albert Joe, and this is called MoCA. And what MoCA does is that it does this multi-omics profiling of toxicity pathways. It looks for the sparsest set of co-regulated genes forming modules, also looking for the sparsest set of co-regulated metabolite modules, and looks to only find those elements and those modules that are associated among the different omics modalities. So in our case, we're doing gene expression profiling using RNA-seq, but we're also doing metabolomics at different modalities. We're looking at the lipidomics, and we're also looking at the polar positive metabolomics as well. So there's really three matrices of omics data that are being compared in this way. And we organize it in a way where we're pulling out those that are specifically associated with the chemicals, and when available as well, those that are specifically associated with an observed phenotypic outcome. And what does that output look like? Well, in this case, again, because of our pilot study, we have different time points. And now what we're doing is that we're actually now comparing the different omics modalities. And the way that MoCA works is that you can find these intra-omics relationships within an omics and time point. We can see the temporal variation of these relationships across the time points, but we can also find at what point do you have the greatest amount of relationship or overlap in the response elements between transcriptomics and metabolomics. So in this case, It just, as an example, it's at time point 3 where the transcriptome and metabolome are most associated with one another. The data is really showing that we're seeing relationships more in terms of early time points in the transcriptomics compared with elements of the metabolomics that are found at later time points. And it makes a lot of sense given how we understand that, of course, the transcriptome gets turned into a proteome, and it's the enzymes and the proteomes that are driving the differences and changes that are happening at the metabolic level. And here what we are able to do then is to identify these modules, find out those ones that are statistically robust, and then begin to compare these modules against the different species in a pairwise manner. In this case, this is being done for Erythroprophis, and we're looking at the modules that were pulled out as the transcriptome modules, however, driven by their associations with identifiable metabolome modules. But here we're just looking at the transcriptome modules, and we find that there's some modules that are identified in Daphnia that are also found in Drosophila, and those are colored here in this table. And then we can interrogate the genes within those models using a gene set enrichment analysis or some sort of pathway enrichment analysis. Here against KEGG, we're able to identify these top 10 KEGG pathways that are co-responsive based on the observations that we've done at the multi-omics level to Erythroprophis in Daphnia and Drosophila. And that's just one example, and I'm looking forward to coming back in another year and showing you the totality of the data set. So in general, what we're trying to do here is that we're trying to demonstrate toxicity by descent. We're trying to say, hey, you know what? We don't necessarily need to rely on tests on mammalian species in order to tell something specific about the threats to humans or other animals, other mammals. There's sufficient biology here that's shared among deeply divergent species, and that biology includes the biomolecular responses of organisms to the chemical toxicities. As long as now we're able to identify those chemical modes of action, hopefully in the near future, we'd be in a situation of regulating substances not based on observing death in one species, but instead of observing their expression of toxicity pathways that are broadly shared among species. I'd like to finish off by looking now more closely at what we're trying to achieve here. I really like, of course, the adverse outcome pathway framework as a means of describing to ourselves and other people what it is that we're doing. Again, we're focusing on this thing called key events, and in particular, these earliest molecular key events that are found early in the pathogenesis of a disease state. And it's quite unlikely that from this data, we're going to be picking up the molecular initiation events. That's where the biology hits the chemistry. You can envision it as in terms of a ligand binding to a receptor, for instance, initiating a cascade of toxicological responses. But as long as we're able to identify these molecular key events that are tied to other key events that are observed in other areas of biological complexity and are predictive of an adverse outcome, I think that we can do a lot by interrogating in general what are these molecular key events and how are they linked to health in organisms, including humans. And in the past, when we've asked the question, is our environment unsafe? I think everybody here would recognize the canary in a coal mine. Back in the 1800s, the coal miners would bring canaries down the shaft. And as long as the canaries were singing, the coal miners knew that the air that they were breathing was safe. Canary doesn't sing. The coal miners know that there's an early warning that they should get out because there's something wrong with the air. Well, those are the old days. Can we envision now or re-envision what sentinel species like Daphnia can do in light of telling us something about hazards through the identification of these molecular key events, especially if these molecular key events are tied to evolutionarily conserved pathways with other organisms, including humans. So I'd like to show you an example of us applying Daphnia as a sentinel species, as an early warning of toxic waters in Europe. So Daphnia, if some of you are unaware of this organism, is a fascinating creature. It's found in all freshwater bodies on all continents. It is a microcrustacean. It's the size of a grain of rice. And it has a lot of features that makes it conducive to doing toxicological tests. The first thing is that they're found in huge numbers, primarily because they reproduce in a clonal manner. Whenever you have them in a laboratory or the conditions are right, every 3 days, a female Daphnia will produce hundreds of genetically identical daughters. And so you can really have quite a lot of material to work on. And here at the bottom are just jars that are being exposed to the river waters that were extracted from the Chaobai River in China, looking to basically identify these molecular key events of Daphnia exposed to these waters. But fascinatingly with Daphnia as well is that when they do get stressed, they switch from producing genetically identical daughters to genetically identical sons, and then mate with sexually receptive females to then produce unique genetically unique hatchlings or sister daughters that are different from their mothers. So again, the biology here is great because of the clonality, we can also dissect away the differences that are not only tied to the chemicals, but the differences that are tied to the genetics. We can get at the genetic variation to susceptibility in populations through experiments using Daphnia. So here's the case study I'd like to share with you. This is sampling 12 sites in Danube River. This was done in collaboration with researchers at UFC in Leipzig. And they have collected the organic extracts. And what we've done is that we've re-diluted them back to the original concentrations. We've exposed our Daphnia to these waters. And then we've done two different things. The first thing is that we've done a chemical fingerprinting of the water, meaning that we've done non-targeted semi-quantitation of the chemistry of the water, basically using mass spectrometry. What we've also done is the bioactivity profiling, so doing the RNA sequencing for the transcriptome, as well as direct infusion mass spectrometry for the metabolome, measuring both polar positive and polar negative metabolites. So in the schematogram, you can see that we're doing the assays, we're doing the chemistry analysis for chemical fingerprinting using LC and GCMS. After a 40-hour exposure of the Daphnia to these waters, we extract the metabolites and RNA from exactly the same tissues. We measure their co-expression of the genes, the amounts of the recognizable metabolites, and then we apply MOCA and other methods in order to reconstruct these modules and correlate the modules as a function of the water chemistries that were measured across these 12 different sites. Here are the 12 sites along the Danube River that were sampled. Just a note that the first site is the one that's the most inland, and the last two sites, D11 and D12, are the ones that are closest to the Black Sea. And this work was done by Xiaojing Li. She was a PhD student in my laboratory and is now a NERC Knowledge Exchange Fellow, which is fantastic because we're very much trying to promote and to advocate these kinds of ways of examining the health of our environments. So in this case, we're just looking at the data using PCA plots. And you can see just from the fingerprinting of the chemistry within the water, we have some outliers. Along principal component 1 and principal component 2, we have the 12th site that is an outlier along principal component 2, and we have the 11th site that is an outlier within principal component 1. When you're looking at the transcriptomics data, you can also see that D12 and D11 are quite distinct from the transcriptomes that were observed when Daphnia was exposed to the other sites. And the same thing goes for the metabolomics data. Again, the metabolomes of the exposure of waters from sites 11 and 12 are clearly distinct from the metabolites from other sites. And so one of the questions that we can now ask is, well, what's so special about these two sites? Once again, using the same kind of approaches, we're looking now at identifying these putative molecular key events and by networks. And so by looking at the modules that emerge from the RNA or the modules that are expressed or co-regulated with polar or a negative positive polar ions, you can see that we're basically getting a number of modules in the RNA, and this is for site 11. We have 11 RNA modules, 19 polar positive modules. These modules contain 19,000 features or genes. The maximum size of the module is 1,243. The minimum size of one of these modules is 20. And this is what they look like. They're kind of like a hairball of co-expression networks. And the question now is, for each of these modules that are putatively molecular key events, is it telling us something about how biology, in this case the Daphnia, is being impacted by exposure to the chemicals as mixtures within these river waters? And so, looking at now at what are the identifiable chemicals as mixtures within those waters that are driving the greatest amount of biomolecular changes, we come up with a list of 8 to 10. And what we're looking at here is two different ways of identifying them. One of them is using the minimum redundancy maximum relevance approach. The other one is using the sparse canonical correlation analysis. And these are being basically compared to the biomolecular data. And here are the pairwise correlations. So metolachlor happens to be the substance that is the most correlated with the changes in the concentrations of this substance as a function of the changes that are happening in the biomolecular responses. And if you look at the top 10, for instance, using a joint correlation, the top 10 here explain 53.3% of all the variation, and there's a correlation among the 10 of 62%. So it's quite high, and it's demonstrating that among the hundreds, if not thousands, of substances that could be found within those waters in the Danube River at site D11, these are the ones that are making site D11 different from all the others. And you can see how, in this case, if you look at the concentrations of these 8 substances across the 12 sampling sites, you can see that these 8 are the ones that are practically non-existent in D11, but are found in higher concentrations in the other sampling sites. And now that is worth investigation unto its own, what's happening at that site for these chemicals. But it was the omics data that pulled out these chemicals, right, as being something that's going on at that site that's making the Daphnia respond differently in a biomolecular way. So we can ask the question now, well, can we identify those metabolites and those genes that are most representative and most explainable in terms of the differences in the biology from D11 against other sites? And so this is using the SCCA modeling for all 8 chemicals that we're looking at. Can we identify the genes, the polar positive metabolites and polar negative metabolites along these axes that basically separate D11 from the other sites? And in this case, the RNA-seq explains 7% of the total variance, and the polar positives 11, and the polar negatives around 8%. So if you pull out now those elements that are most informative of what's happening at that site, they are formed in modules, of course, and these are the modules that come out of this analysis. For instance, there's a module 1 from the RNA-seq data. It has 922 genes. 438 of them are shared with humans. It's significant in terms of their co-expression. And then you can ask the question, are there any pathways that are enriched by these gene sets? And you begin to see and be able to annotate some of the responses that are occurring, not as genes, but as pathways. And here you're basically seeing ABC transporters, you're seeing chemical carcinogenesis, receptor activation happening, you have PPAR disruption of the signaling pathway, drug metabolism, and so forth. So that's for D11. So we now are basically saying we've identified the substances from D11 that are doing something different to the biology of Daphnia, and this is the biology that's being interpreted as being perturbed. And finally, this is what we're doing, right? We're basically associating the chemical set that identified 8 chemicals with the putative molecular key events that came out of the multi-omics data. and are therefore able to interpret those using pathway profiling as being some pretty significant pathways if the interest here is understanding toxicity. And you can see that some of them are in fact labeled here in red. Finally, as we saw before, the 12th site was also different. But in this case, we've identified, I believe, 10 substances for the site 12 that are driving a different biology in the organism compared to the other sites. And in this case, it's because these here are found in greater concentration in this 12th site compared to the other sites. And when you do the same experiment, you're doing the same data analysis, you come up again with the association of 10 different chemicals. Some of them are pharmaceuticals, others are herbicides, others are sweeteners, some of them, and so there's a mixture here of different substances that as and in a mixture are creating these biomolecular changes identified as putative molecular key events, and these are once again interpreted as distinct pathways based on their enrichment of pathway profiles. So let me conclude. I think that what we've seen here is that there's a lot of signal in the omics that can be attributed to the shared biology of organisms, and that shared biology is reflected in terms of applying an evolutionary thinking to understanding and interpreting toxicity. But in terms of using now one of those species as a sentinel, we've been able to show here that the mixture effect of a subset of chemicals under 10 can be distinguished from other sites that are sampled within a river basin, for instance, that the bioactivity profile, the pathways, are indicative of a plausible toxicological outcome from the exposure to this mixture. And these include oxidative stress, genotoxicity, hormone dysregulation. That includes the PPAR signaling pathway that regulates lipid metabolism, glucose homeostasis, inflammation, and so forth. And of course, we were observing that in Daphnia, but keep in mind as well, we're interpreting the gene expression profiles in light of the Daphnia genes as being orthologs to humans, as well as the pathway is being shared between humans and other species. And finally, we were able to investigate the co-expression of these biological features within biomolecular reactions and pathways, and these can reveal the nature of the molecular key events. This permits us to link this data-driven modeling that's being done here with knowledge-based interpretation of the results. And there's no better way to actually have shown that than to use Mentor, for instance, where it's literally being it's using the data to map onto all the known associated functional ties between genes and humans. So finally, can we reimagine sentinel species using multi-omics data to identify environmental hazards? You know, what we've shown here is that you put Daphnia in river waters, they respond. And we're asking Daphnia, is there something wrong with the water? And the answer is yes, and let Daphnia tell us what that is. But because it's also a keystone species, it's already used as a biomonitoring method of ecosystem functioning, for instance. We're just extending that to the biomolecular level, not just observing that something's wrong, but identifying what the root cause of the problem is. But because toxicity can actually be viewed as something that's shared, broadly speaking, across the animal kingdom, including humans, maybe what we're talking about here is not just reimagining sentinel species, but reimagining sentinel species in the protection of both the ecosystem health and human health, and therefore coming up with some crazy notion like One Health toxicology, where ecotoxicology for the protection goals of the environment need not be so different from the human toxicology that has the protection goals of protecting human health and human populations. And this, again, many of you know, this is Precision Tox, but we do this in collaboration now with this large project called PARC, the Partnership for the Assessment of the Risks of Chemicals, where some of you in this call are participating in as well. So I thank you very much for having been there for me to share the results of this investigation.
Thank you, John, for such an amazing talk. I think everybody will agree that it was really dynamic and interesting. And I think you cover the main topics of this workshop, so the complexity of working with omics and also the importance of using a non-model species or other species, because most of the data that we have is human as well. So thank you for that. And now we have a few minutes, so maybe we might have time for a question.